energy is required for normal functioning of all organs of the body, and this energy comes from food by converting fat and proteins into energy for muscles and carbohydrates into glucose. Glucose occupies a central position in metabolism of plants, animals, and many microorganisms. It is not only an excellent fuel but also a versatile precursor capable of providing a huge array of metabolic intermediates for biosynthetic reactions via glycolysis. Glycolysis term is taken from Greek word glykeis, means sweet or sugar, and lysis meaning splitting. Glycolysis was the first metabolic pathway which occurs in the cytosol of the cell with the largest flux of carbon in most cells to be elucidated. The work was initiated in 1897 with Edward Buchner's discovery of fermentation. Till 1930s, as the reactions of glycolysis in extracts of yeast by Otto Warburg and Hans von Euler-Chelpin, and in muscle by Gustav Embden and Otto Meyerhof, were a major focus of biochemical research. It is a process of catabolism in which glucose molecule is oxidized to pyruvate molecule with generation of ATP and NADH via 10 enzyme mediated steps. Pyruvate is then utilized either in aerobic respiration to carbon dioxide via TCA cycle, or in anaerobic respiration it is fermented to lactate or ethanol. More energy is derived from glucose in both of these processes through the formation of ATP. There are two main phases of glycolysis: investment phase and payoff phase. Each involves 5 steps. First phase is investment or priming or preparatory phase. This phase of glycolysis requires ATP. In this phase, 2 molecules of ATP are invested and the 6 carbon chain is cleaved into 2 triose phosphates. This phase involves 5 steps which are as follows. First step, phosphorylation of glucose. The first step of glycolysis is irreversible phosphorylation of D-glucose to glucose 6-phosphate G6P, as the 6th carbon of glucose acquires the phosphate group, and in this process 1 ATP is consumed. This process is mediated by enzyme hexokinase, which requires Mg2+ for its activity. It shields negative charges of phosphoryl groups in ATP, which makes the terminal phosphorus group an easier target for nucleophilic attack by hydroxyl group of glucose. Second step is conversion of glucose 6-phosphate into fructose 6-phosphate. Rearrangement of aldose glucose 6-phosphate G6P into a ketose fructose 6-phosphate F6P by reversible isomerization reaction mediated by glucose phosphate isomerase. In other words, phosphoglucose isomerase, which also requires Mg2 plus for its activity. Third step is phosphorylation of fructose 6-phosphate into fructose 1,6-bisphosphate. In this step, fructose 6-phosphate is converted to fructose 1,6-bisphosphate FBP in the presence of enzyme phosphofructokinase PFK. Second molecule of ATP provides the phosphate group that is added onto the F6P molecule. Similar to the first step, Mg2 plus act as a cofactor and is involved to help shield negative charges. Next is the fourth step, which is cleavage of fructose 1,6-bisphosphate. This step utilizes the enzyme aldolase, which catalyzes the cleavage of FBP to yield 2 3-carbon molecules. One of these molecules is called Glyceraldehyde 3-phosphate GAP and the other is called dihydroxyacetone phosphate DHAP. Fifth step, which is interconversion of triose phosphates. Glyceraldehyde 3-phosphate GAP is the only molecule that continues in the glycolytic pathway. As a result, all of the dihydroxyacetone phosphate DHAP molecules produced are further acted on by the enzyme triphosphate isomerase TIM, which reorganizes the DHAP into GAP so that it can continue in glycolysis. At this point in the glycolytic pathway, we have 2 3-carbon molecules, but have not yet fully converted glucose into pyruvate. So, here comes the next step, which is payoff phase, in which some of the chemical energy of the glucose molecule is conserved in the form of ATP and NADH. Sixth step is the oxidation of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This is the first step of payoff phase, which involves oxidation of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate mediated by enzyme glyceraldehyde 3-phosphate dehydrogenase GAPDH. The enzyme GAPDH holds the molecule in a conformation such that it allows the NAD molecule to pull one hydrogen off the GAP, converting the NAD to NADH. The phosphate group then attacks the GAP molecule and releases it from the enzyme to yield 1,3-bisphosphoglycerate, NADH and a hydrogen atom. Now, the seventh step, which is phosphoryl transfer from 1,3-bisphosphoglycerate to ADP. In this step, phosphoglycerate kinase PGK transfers the high energy phosphoryl group from carboxyl group of 1,3-bisphosphoglycerate to ADP forming ATP and 3-phosphoglycerate via substrate level phosphorylation. Mg2 plus is involved to shield the negative charges on the phosphate groups of the ATP molecule. Now, the 8th step, which is conversion of 3-phosphoglycerate to 2-phosphoglycerate. This step involves a simple rearrangement of the position of the phosphate group on the 3-phosphoglycerate molecule, making it 2-phosphoglycerate using phosphoglycerate mutase PGM. The reaction mechanism proceeds by first adding an additional phosphate group to the 2' position of the 3-phosphoglycerate. The enzyme then removes the phosphate from the 3' position, leaving just the 2' phosphate and thus yielding 2-phosphoglycerate. In this way, the enzyme is also restored to its original phosphorylated state. Now, the 9th step, which is dehydration of 2-phosphoglycerate to phosphoenolpyruvate. This step deals with conversion of 2-phosphoglycerate to phosphoenolpyruvate PEP in the presence of enzyme enolase, which involves irreversible removal of water molecule from 2-phosphoglycerate. And the last step, which is the 10th step, is transfer of the phosphoryl group from phosphoenolpyruvate to ADP. The final step of glycolysis is the transfer of phosphoryl group from phosphoenolpyruvate to ADP, catalyzed by enzyme pyruvate kinase, which requires K plus and either Mg2 plus or Mn2 plus for the activity. As the enzyme's name suggests, this reaction involves the transfer of a phosphate group. The phosphate group attached to the 2' carbon of the phosphoenolpyruvate PEP is transferred to a molecule of ADP, yielding adenosine triphosphate and pyruvate, the end product of glycolysis. Again, since there are 2 molecules of PEP, here we actually generate 2 ATP molecules. Balance sheet is constructed to know about the fate of carbon skeleton of glucose and input of inorganic phosphate and ADP with output of ATP, that is adenosine triphosphate. And finally, the pathway of electrons in the oxidation reduction reactions. Left hand side of the reaction shows input, while the right hand side reactions show output of the reaction. The reaction is glucose plus 2 ATP plus 2 NAD plus 4 ADP plus 2 inorganic phosphate yields 2 pyruvate plus 2 ADP plus 2 NADH plus 2 H plus ion plus 4 ATP and 2 H2O. On cancelling common terms from both sides of the equation gives the overall equation for glycolysis under aerobic conditions, which is glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate gives 2 pyruvate plus 2 ATP plus 2 NADH plus 2 H plus and 2 water molecules. The 2 molecules of NADH formed by glycolysis in the cytosol under aerobic conditions undergoes electron transport chain in eukaryotic cells. Energy transfer from NADH to oxygen in mitochondria provides energy for synthesis of ATP, adenosine triphosphate, by respiration linked phosphorylation. With exception of some bacteria, the pyruvate formed by glycolysis is further metabolized via one of the 3 catabolic routes. In aerobic organisms or tissues under aerobic condition, glycolysis is the first stage in complete degradation of glucose. The end product of glycolysis, the pyruvate, is oxidized with loss of its carboxyl group as carbon dioxide to give acetyl group of acetyl coenzyme A. The acetyl group is then oxidized completely to carbon dioxide by citric acid cycle. Electrons from these oxidations are passed through a chain of carriers in mitochondria to form water. The energy from electron transfer reactions drives the synthesis of ATP in mitochondria. Second route for pyruvate is its reduction to lactate via lactic acid fermentation. Vigorously contracting skeletal muscles under hypoxic conditions reduce pyruvate to lactate, accepting electrons from NADH and thereby generating NAD plus necessary for glycolysis to continue. Certain tissues and cell types like retina and erythrocytes convert glucose to lactate even under aerobic conditions, and lactate is also the product of glycolysis under anaerobic conditions in some microorganisms. The third major route of pyruvate catabolism is ethanol or alcohol fermentation. In some plant tissues and in certain invertebrates, protists and microorganisms such as brewer's or baker's yeast, pyruvate is anaerobically converted to ethanol and carbon dioxide. In type 1 diabetes, which is the insulin dependent diabetes, patient undergoes accumulation of high levels of glucose in the blood, a condition which is known as hyperglycemia. Defective uptake of glucose by muscle and adipose tissue possesses profound effect on metabolism of carbohydrates and fats. Also, in many type of tumors found in humans and other animals, glucose uptake and glycolysis proceed about 10 times faster than in the normal non-cancerous tissues. The high glycolytic rate in tumor cells also has diagnostic usefulness to pinpoint the location of tumors. The phenomena of aerobic glycolysis increase in cancer cells was first described by Otto Warburg in 1930. and hence this is the better known as Warburg effect. According to Pellicano et al. In 2006, development of novel glycolytic inhibitor may work promisingly as anti-cancer agent. Glycolysis is an almost universal central pathway of glucose catabolism, in which a glucose molecule is oxidized to two molecules of pyruvate with energy conserved as ATP and NADH. Glycolysis is tightly regulated in coordination with other energy yielding pathways to assure an adequate supply of ATP.